Hello, this is Dr. Rajesh Yadav, PhD from All in Institute of Medical Science, New Delhi, India. Today we will discuss about the topic muscles contraction. So first of all, we should know about the structure of myosin filament and active filament. Because in the muscles contraction, actin and myosin filament slide to each other. So when we take heavy things, at that time the muscles contraction occurs, where the role of actin and myosin molecule. So first of all, we should know about the structure of myosin filament. So myosin filament is made up of myosin molecules. So one myosin filament is made up of 200 molecule of myosin molecule. So the figure A is showing the structure of the myosin molecule. So one myosin molecule is made up of 6 polypeptide chain. Out of 6, 2 polypeptide chain form tail of the myosin molecule and it is called heavy chains, while the 4 chain of 6 polypeptide molecules form head of the myosin molecule and these are called light chain. So the figure B, which is showing that structure of myosin filament. So these myosin molecule form myosin filament. So the tail of the myosin molecule form the body of the myosin filament, while the head region form cross bridge and hinge region of myosin filament. So again, if we discuss about the important thing about the myosin filament, that these head form the myosin cross bridge. So the cross bridge contain ATPase enzyme activity. So the important point of this cross bridge is that it has ATPase enzyme activity. And during the muscles contraction, the ATP bind with the cross bridge or myosin head. As a result, these enzymes break down ATP into the ADP and phosphate. As a result, energy release, and this energy will help for the contraction of the muscles contraction. And this energy will be responsible for the tilting of the myosin head. So now we will discuss about the structure of actin filament. So actin filament basically have 4 components. One is F-actin, second tropomyosin, third troponin complex, and fourth active sites. So first we will discuss about the F-actin. This F-actin is made up of double helical structure and it is from double helical structure like the DNA. So each strand of double F helix is composed of polymerized G-actin molecule. So if we see in the figure, this F-actin is present in the light pink color and there is one chain of the uh, F-actin and this is the second chain of the F-actin. So both light chain is F-actin and on the F-actin the G-actin molecule is present and this G molecule is considered as ADP. So it is from the active sites of the actin filament. So the second things about the tropomyosin. We have discussed about the F-actin, which is two spiral chain which is spiral to each other. Now come to the tropomyosin. Tropomyosin has molecular weight 70,000 and length 40 nanometer. These molecules are wrapped spirally around the site of the F-actin. So in the figure, this dark chain is called tropomyosin, which is spiral with F-actin chain. So in the resting state, the tropomyosin lie on the top of the active sites of the active strands, so that contraction cannot occur between the actin and myosin filament. Now third structure is a troponin complex. Troponin is a third molecule which is present on the active chain, active filament, and it has basically 3 sites. One is troponin I, second troponin T, and third troponin C. So troponin I is responsible because responsible for the attraction because it has a strong affinity with actin filament, while the troponin T has affinity with the tropomyosin molecule, while third troponin C has affinity with the calcium ion. So the complex is believed to attach the tropomyosin to the active, and the strong affinity of the tropomyosin for the calcium is believed to initiate the contraction process. Now we'll discuss the mechanism of the muscles contraction. So in the first step, first of all, when the action potential reaches to the neuromuscular junction in the muscles, sarcoplasmic reticulum is present. So as the result of action potential, sarcoplasmic reticulum release calcium ion in the cytoplasm. As the calcium ion reaches the troponin C binding site, it will bind with the troponin C and the conformational change in the active filament. As a result, the active site G molecule will expose. In the second step, the myosin filament, which is from the cross bridge, we have already discussed that it has ATPase enzyme activity. So when the ATPs bind with this ATPase enzyme, so ATPase enzyme break down ATP and release the energy. As a result, this energy is responsible for the tilting of this cross bridge. So as a result, this cross bridge tilt in the forward direction, and this phenomenon is called power stroke. So after the tilting of the cross bridge or myosin head, this ADP and inorganic phosphate again combine and form ATP, and this ATP bind with this another cross bridge molecule and repeat the same phenomena. As a result, again ATPase enzyme break down ATP and energy will be released, and this energy will be responsible for tilting of the myosin head. As a result, power stroke occur, and after the tilting, ATP and inorganic phosphate combine form the ATP, and that will bind with the another cross bridge. So in the this process will repeat again and again, and in this way muscles contraction occurs. So this is the mechanism of muscles contraction. So we will summarize in the next step. So as we have discussed that we need large number of ATP which is responsible for the muscles contraction. So if we are doing heavy work, so large number of ATP will be required there, and this phenomenon is called fan effect. So in the first step here, we have already explained that first of all action potential reach and ATP bind with the myosin head, and in the second step tropomyosin troponin complex bind with the calcium ion at the active site of the actin filament, and active sites are uncovered. In the third step, the cross bridge interact with the active site of the actin filament. As a result, power stroke occurs and ATP will be breakdown and tilting of the myosin head. Once the head of the cross bridge tilt, it allows release of the ADP and phosphate that were previously attached to the head. At the site of release of ATP, a new molecule of ATP bind, and the binding of new ATP causes detachment of the head from the actin. After the head detached from the actin, the new molecule of ATP is cleaved to begin the next cycle, leading to new power stroke. That is, the energy again clock the head back to the perpendicular condition, ready to begin the new power stroke cycle. When the cocked head binds with the new active sites of the actin filament, it becomes uncocked and once again provides a new power stroke. So this is the mechanism of muscles contraction. Thank you.